Welcome back to Grad Diaries. In this video, we are going to understand DNA transcription, one of the most important processes in molecular biology. We'll look at what transcription is, major factors involved in it, and how the process occurs step by step from initiation to termination. Before understanding transcription, we need to understand what central dogma of molecular biology is. DNA, which stores hereditary information in a sequence of its bases, it forms RNA through the process of transcription, and RNA is processed to form protein, is called the process of translation. The genetic information flows from DNA to RNA by the process of transcription, and RNA to protein with the process of translation. And in this video we are going to understand what transcription is. So what is transcription? Transcription is the biological process by which segment of DNA is used as a template to synthesize a complementary RNA molecule, which is known as messenger RNA. What happens in this case is transcription is the first process which helps to form the functional protein, and in case of eukaryotes this process takes place in nucleus where DNA is stored, and in case of eukaryotes it occurs in cytoplasm. This RNA is then exported to cytoplasm for translation, and this process happens in the flow of genetic information, which is DNA to RNA, and which ultimately forms protein. And this is the central dogma of molecular biology. So now let's understand what are the key factors involved in the process of transcription. First and foremost is RNA polymerase. This is the core enzyme involved in synthesis of RNA using DNA as a template. That is why these polymerases are also known as DNA dependent RNA polymerase, which means it uses DNA as a template and synthesizes RNA. What RNA polymerase does is it unwinds double helix DNA and catalyzes the synthesis of RNA by adding ribonucleotides in the direction of 5' to 3' direction. The next factor is transcription factors. These are the proteins, regulatory proteins, which control different processes, different steps of transcription from initiation to termination. And the general transcription factors involved are TF2, D, H, A, B, etc. These factors assemble at the DNA to form the pre-initiation complex, which recruits RNA polymerase and undergoes the process of transcription. Regions of DNA like promoter region and other regulatory elements, these are DNA sequences which define the recruitment of transcription factor site, transcription start site, and others regions such as enhancers, silencers, etc. So this is the overview of transcription process, which is initiation. In this process, transcription factors bind to the region of DNA that is known as TATA box, and there RNA polymerase is recruited, which forms pre-initiation complex, and the DNA double helix is unwound to expose the template strand, enabling the transcription to begin. Next is the elongation. What happens in elongation is RNA is being formed and extended in the along the DNA template in the direction of 3' to 5' direction. Using DNA template, which is from 3' to 5' direction, it synthesis RNA in the direction of 5' to 3', and nucleotides are added on the 3' end, which is the growing end. The third step is termination, where RNA polymerase identifies a terminator sequence, or in case of prokaryotes it may be rho factor or sometimes hair loop structure. It stops the RNA synthesis and cleaves the generated RNA. And then comes the RNA processing. In eukaryotes, the pre-mRNA undergoes processing such as 5' capping, which protects the transcript, and splicing, which removes intron via the spliceosome, and polyadenylation, which is the addition of poly A tail at the end of 3' end. These processing forms mature mRNA, which is being transported from nucleus to cytoplasm for translation. A DNA transcription is a tightly regulated multi-step process, and each step have a particular process. Suppose in initiation we form the pre-initiation complex, then elongation, followed by termination and processing of RNA. Steps are coordinated by multiple molecular factors which ensure accuracy and efficiency of the RNA molecule for protein production and cellular function. Let's see each step separately. Let's understand what happens in initiation. In the promoter region, there is a segment of DNA called TATA box. This segment of DNA recruits transcription factors in an order as shown here. At first, TF2D is recruited, which have a TBP domain, which is TATA box binding protein domain. This is followed by TF2A and TF2B is recruited to the TATA box site. Upon this, other factors like TF2F with RNA polymerase is recruited, then TF2E and TF2H is recruited to the TATA box site. Once these factors are recruited, RNA polymerase unwinds the DNA and exposes the template strand, which is 3' to 5' for the RNA synthesis. Now this is initiation complex and is now ready for the RNA synthesis. Now what happens in elongation? Now that we saw that RNA polymerase have unwound the DNA, it uses the template strand 3' to 5' to form the RNA from 5' to 3' direction. And the direction RNA polymerase moves is 3' to 5' direction of the template strand. RNA polymerase reads DNA in the 3' to 5' direction. RNA synthesizes from 5' to 3' direction. And in the process of synthesis, ribonucleotides are added at 3' end of the growing RNA. And this happens as follows. In place of template strand, if A is present, converted to U. In place of T, A will be coming C, G and G, C. Now let's see what happens in case of termination. So there are two types of transcription termination mechanism in case of prokaryotes: row independent termination mechanism and row dependent termination method. So what happens in row independent termination is that as RNA polymerase moves forward, RNA is being synthesized, and at a point it finds a terminator sequence, forms a hair loop structure, and this sequence is followed by poly U. So once these hairpin RNA is formed, which dissociates RNA polymerase, and RNA is released. Now in case of row dependent, as RNA polymerase moves forward, it identifies a RUT site at which row protein binds. So once it binds to that factor, it kind of signals the RNA polymerase dissociation and release of complete RNA from there. Now in case of eukaryote, what happens is as the RNA polymerase moves, RNA is being synthesized, and at a point it identifies adenylation site in RNA, which kind of signals the cleavage of pre-mRNA to form the mature RNA and release of RNA polymerase 2. Now RNA processing. How the RNA is processed to form the mature mRNA. Now as we saw, gene is being synthesized to pre-mRNA with the help of RNA polymerase 2. This RNA is then processed to add 5' cap, which have 7-methyl guanosine, and it is known as 5' capped pre-mRNA. This again undergoes splicing with the help of spliceosome, removes intron and attaches the exon to each other. It is then followed by addition of poly A tail at 3' end. Now this This is actually the mature mRNA which is being transported out of nucleus in the cytoplasm for the formation of protein. Now let's summarize what we have studied. Transcription is the foundation of gene expression, enabling the synthesis of RNA from DNA template. The process drives the production of proteins essential for cellular function and biological processes. DNA transcription is a regulated process involving transcription factors, DNA elements such as TATA box, and other regions of DNA such as enhancers, silencers. Transcription forms the molecular basis for the cellular function, as it forms RNA and it is then converted to form protein. Unregulated transcription process may lead to numerous diseases and can be a target for therapeutic research. Thank you.